These are the lecture notes for Chapter 3 on cells. And this is a picture of a cell. It's kind of cut open so you can see inside of it, and you can see all the cell parts. You may have made a model of a cell when you were a kid in science class out of Play-Doh, or I've had students make models of cells out of cake. But our body is made of trillions of cells, and they don't all look like this one. But this one is just used to show you, like a typical cell. All right. The main parts of a cell are going to be the outside of it, which is like the skin, is called the plasma membrane. But it's not really like skin so much because it's more of a fluid. It seems like it would be a solid, but it's not. It's more of a fluid. And the molecules in the plasma membrane, they don't stay in place. They move around. So it's a fluid type of structure. The nucleus is inside the cell, and it's also enclosed in a membrane, and it contains the DNA and pieces of DNA called genes are what code for our traits. And then the cytoplasm is the fluid inside the cell, but it's actually defined as the space between the plasma membrane and the nucleus. So it's like between the outside of the cell and the nucleus of the cell. And inside of the cytoplasm, you're going to have organelles floating around within fluid called cytosol. So let's see if that's on the next page. No. Okay. So I'm going to add it. So cytosol plus organelles together equal cytoplasm. All right. The next slide shows you or goes through different functions of the cell. We're going to skip that. And then this is a typical human cell. There's a section on microscopes, by the way, but that's going to be covered in lab. So I'm going to move on from microscopes. Then we get to section 3.3. And I am kind of moving fast through the sections because this is a huge chapter, and you will even notice, and I go ahead and apologize in advance because the homework assignment for chapter 3 is very long, and I try to keep it, any homework assignment that I give, I try to keep the amount of questions around 40. So sometimes for the longer chapters, you're going to have 50 or more, but this one is actually 68. And I went over it many times, and I deleted the ones I felt like I could delete. But the questions that are left in there are really good questions for reviewing for the TEAS test. And just the chapter is just a very long review chapter. So anyway, I apologize for that. I won't do it to you every single week. Functions of the plasma membrane. Well, it functions as a boundary. So it encloses and supports the inside contents of the cell. It also attaches the cell to its surroundings, and it's able to recognize and communicate with other cells by binding to them and recognizing chemicals, that kind of thing. And the main thing is the plasma membrane determines what substances will move into the cell and what substances will move out of the cell. So what we're looking at right now, I'm going to blow up so you can see it a little better, is a model of the plasma membrane. The main component of the plasma membrane are phospholipids, these little blue, they almost look like blue balloons with two strings hanging down instead of one string. But they are phospholipids, and the phospholipids are oriented in such a way that the phospholipid heads, which are blue, these structures are going to face outside the cell to the extracellular fluid and inside the cell to the cytosol because they are water soluble. But the two little tails that are attached to each phospholipid, they're going to face each other because they are not soluble in water. They're insoluble in water. They are lipids. So they're going to face each other and not face the watery environment inside or outside the cell. You also see these very large molecules that go all the way through the cell, and they are large proteins and they have different functions. Like this one, for example, has a channel that allows molecules to move in or out. Any of the structures that you see with these long yellow chains, chains of these yellow hexagons, the yellow hexagons are they're actually glucose molecules that are attached into a chain. So they're polysaccharides is what they are, but they are carbohydrate chains, okay? And the carbohydrate chains kind of make the outside of the cell sticky, so they sort of help. They're called the glycocalyx of the cell, and they just sort of make it sticky so it can stick to it can stick to other cells and it can stick to its surroundings. But they also help other cells, like cells of the immune system, to recognize our own cells. They're markers. Sometimes, like when we get to the chapter on the immune system, these markers are going to identify, for example, whether or not these cells are bacterial cells or whether they're foreign cells of some kind that might be dangerous. They will tell our immune system to attack them and attack anything else that looks like them. But these kind of markers also mark our tissues, and we call them tissue compatibility markers. And so if you are wanting to donate an organ to someone, say one of your relatives needs a kidney and you decide to donate your kidney, well, you have to get a tissue compatibility test. And if all of these markers match, then you're a tissue match. So that's just some of the functions of these carbohydrate chains. If they're attached to a protein, we call them a glycoprotein. And if they're attached to one of the phospholipids, we call it a glycolipid. But that's what they are. They're markers. And then you see cholesterol, these little yellow 4-ring structures that you see in here embedded within the phospholipids. What the cholesterol will do is, I told you that the plasma membrane is kind of a fluid structure, and all of these molecules that you see in this picture, they're actually able to move past one another. I mean, they don't just stay there. They don't jump around, but they scoot around. They kind of scoot past one another. Like if you could imagine a box full of marbles and you just sort of tilt it, the marbles are going to move past one another, but gently. They're not going to jump around. Well, that cholesterol makes the membrane thicker. It makes it not as fluid. It makes it more solid. Okay, so it gives it more consistency. So we do need cholesterol in our diet, in case you were thinking that we don't and you're trying to do low cholesterol. You can do low cholesterol, but don't ever do no cholesterol. All right, so the lipids in the plasma membrane are phospholipids and cholesterol. So make sure you know that the plasma membrane primarily is composed of a phospholipid bilayer, a double layer of phospholipids. The heads of the phospholipids are polar. Remember, polar, that water is also polar. So they're going to face the watery environment inside and outside of the cell. The word that we use for this is hydrophilic, meaning water-loving. The polar heads are hydrophilic. And then the non-polar tails, the tails are actually lipids. They face each other and they face away from the watery environment inside and outside the cell. So we call them hydrophobic. Hydrophobic means fear of water. Hydrophobia is actually rabies. And so it's the medical term for rabies. So hydrophobic is fear of water, because that's one of the symptoms. And that just doesn't mean that the nonpolar tails are afraid of water. It just means that, they are insoluble in water. The cholesterol that is scattered, it says interspersed or scattered among the phospholipids, determine the fluid nature of the membrane. So they provide stability to the plasma membrane. They make it more solid and more stable. All right. The different proteins, there's about 6 different types of proteins found within the plasma membrane. And these ones that have these carbohydrate chains attached to them, they're marker molecules. So we're looking here at a glycoprotein or a cell surface marker. If it's a glycoprotein or a glycolipid, they're going to allow cells to identify themselves to another cell. Then there are proteins called attachment proteins. They help attach the cell to other cells. They help attach the cell to other components inside the plasma membrane and outside of it. So if they're called cadherins, they will attach cells to other cells, cadherins. And if they're called integrins, they work in pairs to attach the cell to extracellular molecules, to molecules outside the cell. So there are attachment proteins, there are marker molecules, there are transport proteins. These that you're looking at here are called carrier proteins. What they do is they chemically bind to a certain substance and bring it across the membrane. They can bring it into the cell or out. So they're called carrier proteins if they chemically bind to a substance and carry it across. But they're called channel proteins if they just form an open channel for that substance. There is a section on ion channels, but we go over that anyway when we get to muscle contractions. So make sure that you know that some carrier proteins only carry one substance, and so they're called uniporters. They only move one substance. So like maybe a carrier protein specifically just for sodium. But then there's symporters. And what they do is move 2 substances in the same direction. A very common symporter in our cells is one that transports sodium into the cell and glucose into the cell along with the sodium. So it's a type of transport called co-transport because you're transporting 2 molecules in the same direction. And if it's an antiporter, it's going to transport 2 molecules in opposite directions. One will come into the cell and one will move out, and that's called counter-transport. When we learn about the sodium-potassium pump, then we will talk about counter-transport. ATP-powered pumps are proteins that have to bind to ATP in order to pump a substance in like the opposite direction of its concentration gradient. Instead of the substance moving from where its concentration is high to where it's low, it's going to move in the opposite direction. It's going to move toward its high concentration. Okay, so ATP is the molecule that our cells use for energy, and this is one of the purposes, one of the functions of the ATP is powering these pumps. Then you have receptor proteins, and a receptor typically will bind to a chemical like a hormone, and whatever that chemical is that binds to the receptor, we call it a ligand, okay? So whether it's a hormone or an enzyme or just, I don't know, anything, whatever chemical it is, we call it a ligand, whatever that chemical is that binds to the receptor protein. And then once it binds, it's like the key to triggering some kind of change inside the cell. For example, there's a hormone called insulin, and when insulin binds to its receptor on a liver cell, it causes the liver to bring glucose into the liver cells and store that glucose, and in that way it can help lower your blood sugar, lower your blood glucose levels. These are gated channels that bind to ligands, but I'm going to skip this. Okay, now we're on the section called enzymes, and this is slide number 29. And so I'll stop here, and we will start the next recording with enzymes.